Good afternoon. Thanks for Trevor and Marcus for the invitation. Thanks for the person that canceled last minute. I hope he or she is doing well. But I'm grateful for the opportunity. And I'll be telling you about our work on combinatorial recognition by bZIP transcription factors. And this is work I did in a Sims lab with the BIRS doing all the computational work. Don't be fooled by a sim holding the pipette. I did all the pipetting. And this was a great collaboration with Amy Keating and Aaron Reinke, who's now in Toronto setting up his lab. And this was published last year. Okay, so the main motivation is how we go from one genome to so many phenomes and responses in multicellular organisms. And a current model is differential use of the genome. And this differential usage is driven mostly by transcription factors, which have been cataloged and now at how many? 1,639.
It's 41 now, right?
I'm missing two.
Yeah. No, that's from the talk.
So, and we're focusing on bZIPs in humans. Is the third largest family, and bZIPs bind DNA in this scissor grip style. And here I'm showing you Chuck Binson's model prediction around 3 years before the first structure came out for GCN4. You have a losing zipper for dimerization domain, and then you have basic alpha helices inserting in the major groove of DNA, making the recognition. And this is just in more detail of how those contacts are made in the major groove. And bZIPs are obligate dimers. They can bind, as been shown in many presentations here, as either homodimers or heterodimers. So this poses the paradigm of benefiting from combinatorial regulation by these factors. So we were asking if the DNA specificity of heterodimers is different from homodimers, whether these heterodimer sites are being bound in cells, and whether we can predict impact of binding for SNP variants. So there are 54 bZIPs. So if you take into account all possible combinations, this can, even for a 50-something member family, the number of motifs that they can end up recognizing is pretty large. So to guide which of those dimers to test, we were guided by work done by Amy and Aaron on protein-protein interactions by this family in absence of DNA. In this case, out of the 54, they selected 36 bZIPs that cover the diversity of sequences of the family, and then tested all possible 666 combinations, and around a third of those stably associate in absence of DNA, and stably associate is with KDs under micromolar. So of those, we selected around 300 bZIP dimers to test, including all 36 homodimers and the rest heterodimers. And you can tell from the blue dots that it's not even coverage of the matrix. It was biased mostly. We selected members that did not homodimerize, so that we could take advantage of that and use that factor for pull down to at least try to enrich for heterodimers. So we did this by HT-SELEX. And we had our 20 base pair library in this experiment. And one of the bZIP partners was biotin labeled. And it was either a partner that did not form homodimers in the absence of DNA, or if both form homodimers, we selected the one with the least affinity of homodimerization. We performed a total of 3 rounds of selection and sequence. So the way we analyze our sequencing data, we use sliding windows to count all possible k-mers, and to correct for our bias in our library, we use the 5th order Markov model as in the Slattery et al. Paper. So we did 36 homodimers. We got specificity data for 22 of the 36, and it was consistent with which ones form homodimers in solution. And we retrieve the known homodimer motif, the TRE site, CRE, the cat box, and so on. When we look at heterodimers, the diversity of sites increased as we expected. The majority of those sites were just classical bZIP rules in which you had abutted half sites. And we have a set of those, also very known in bZIPs, in which the abutted sites were overlapping by one base. We call those a variable spacer. And we got a set of which we call the emergent sites. And those were the ones we could not, were not the combination of just joining the preferred sites by the homodimers. And like around 2/3 were cojoined half sites, and 20% emergent and variable spacer. In terms of what we used to guide our experiment, which was dimerization affinity, so most of the interacting partners in the absence of DNA bound DNA specifically. We were surprised about the non-interacting partners that were bound DNA specifically. Around 20% of the non-interacted partners we tested. When we did this, we were super excited. We tested 300 heterodimers. By then, only Richard Mann's the latent specificity paper and Trevor's the NF-kappa-B dimers. So we were super excited that we had 100 heterodimers, and then Asim comes from Banff in Canada and says, oh, Jussi just did 10,000 hetero. So, but since we focus on this family, we learn a lot of insights from bZIP family, which I'll be sharing. We also cluster like the high affinity sequence, and I'm highlighting the usual suspect, the known bZIP sites, but also want to highlight specificity spaces that are only for heterodimer, or are preferred by heterodimers, like this region up here, which is the joint site of CRE sites and cat sites. And I'll be showing more examples of those. In terms of the question whether with combinatorial binding can we expand the specificity, in the case of HT-3 homodimer, which is a repressor, and it has been reported when heterodimerizing with other partners to be an activator. So we got specificity for this HT-3 containing heterodimers, some of which bind the same sites as the homodimer. But as you move down the list of heterodimer, you start finding the cojoin sites with the CVP family bZIPs, and this, which we're calling the emergent sites, when heterodimerizing with the BATF family, which was surprising to us because BATF family members, BATF3 was the only one we got homodimer data for, binding the Cre site, and then BATF with June typically preferred the AP1 site or the Cre site, as was also shown by Gary recently in complexes with IRFs. But in our case, BATF heterodimers with ATF3 factors prefers this asymmetric site, which I still don't know, at least structurally, don't know how to interpret this and not binding the palindromic site. Okay, so this is in like high affinity sites or motif. If you look at the scatter plots, and you have on the bottom the homodimer specificity with the preferred sites colored in orange and magenta, you see them that the homodimer sites have very low predicted affinity for the heterodimer, which is shown here in red, with high affinity site for the cojoined site. So it's hard to look here in scatter plots to find interesting sites or patterns. So in the Ansari lab, for that we use sequence specificity and energy landscapes, in which we display the entire K-mer affinities as a series of concentric rings, which as I mentioned the other day. Just to remind you, in the innermost ring, we display all sequences that have a match for a bZIP motif, can be the highest affinity or any other sequence. Alongside, in this case, we're using 10-mer data and using an 8-mer bZIP, so we get information on the contribution of the bases flanking that bZIP motif. Moving out, we'll display all sequences having one Hamming distance from the bZIP motif, and the way we organize is clockwise, starting with a mismatch at the 5' position and moving along all the way to the 3'. And just as an example, this is the blue. This blue region here will have all mismatches, but it will, since it's an 8-mer seed on 10-mer data, it will also have information on the flanking bases to that site in particular. What's really neat is, since it organizes the data by position and by the flanking, the identity of the flanking, related sequences will pop up, even if they might be buried in the lower part of the scatter plot. It's an example, in this case, June ATF3. June and ATF3 homodimers bind the Cre site, so assuming, this is without looking at the heterodimer data, we assume it will bind the Cre site as well, and draw the landscape with that bZIP motif, and you start seeing the variation on the height of the sequences there that will inform of the effects of flanking sequences, but also related sequences that are all bound with high affinity or mid affinity will pop up as peaks. You can either go manually and check what that sequence is, but in the latest incarnation of SELs, there's an algorithm to pick up peaks in different rings and tell you what relate those sequences. So going back to the homodimer, this is an example of the innermost ring using the Cre bZIP motif. And the reason it looks so sparse, this is using 12-mer data. There's less coverage, but still there's information to be gained, like what are the highest sequences, the highest affinity sequences, and which ones containing the SID motif might be low. So we can go and select the highest affinity flanking for the Cree site and get its apparent KD, and it's around 3 nanomolar. And I messed the order, but if you take the 2 worst flanking sites, you lose like around 2 orders of magnitude in KD. If you have 1 bad flank and 1 good flank, then you get a medium or an affinity in between for those. There's been, Polly got asked, and then during dinner earlier we were discussing, there's always this debate, what's flanking, what's core, where do you stop? In our case with bZIPs, we're calling the Cree site the 8-mer, the SID motif. And to the way I presented, at least on the available crystal structures, no base-specific contacts are made beyond that 8-mer, but with the caveat that crystal structures use as short as possible peptides. But then we ask what SID bases outside the core might be doing. We look at, I don't know if this is ORCID 1 or 2, but you see that variation in the flanks of the 8-mer sometimes have impact that propagates to the first or second bases of the core in terms of ORCID or minor groove, but then other parameters as well. Okay, so the question, so what again, why should we care? Are these sites bound in living cells? For that, we look at chip data from ENCODE. So there are a few cell lines in which a couple of bZIPs were chipped. So in this case, this is a chip trace of ATF3 and CBP beta, binding what we call a heterodimer site, the combination of those sites, and then we use our in vitro intensity to score that loci using a moving window and just plotting our intensity. And you can see that it scores higher than the heterodimer scores higher than either homodimer. And this is, it's not calibrated for specific KD. No, this is just like Gary mentioned the first day, this is the slice along that specificity along the binding curves. So we took these 2 chip-seq data sets and look at the overlapping sites and scored them with the homodimers. We're doing all because the chip is with CBP beta. We don't have CBP beta. So we score with these homodimers. This is CBP beta from Joel Maral, and with our 3 heterodimers. And you can see that distribution shifts to higher intensities when using the in vitro specificity. So at least some of those peaks in the intersection we think might be bound by heterodimers. So the motivation when doing this work, specifically for bZIP, is if you chip ATF3, in principle you should chip down all the ATF3 bound peaks, be it homodimer or heterodimers. So what we were trying to do, having several heterodimers, is to see if we could parse out those peaks on whether it's bound by homo or heterodimers. So we tried doing what Ashley mentioned before, doing the ROC curves, and then using either the ATF3 or combining all our heterodimer data into a data set and score, but none of those seemed to work. What we ended up doing is taking, in this case, K562, this is around 10,000 chip-seq peaks from ENCODE. And so we calculated we made a heat map of the false positive rate at which we stopped detecting that peak as a positive with the in vitro data set below. So for example, all this red, color red, it's very low false positive red rate for the ATF3 in vitro data. And then when we added our the other heterodimers, we see that some peaks are called in common, but then you start seeing patches of peaks that are best called using specific heterodimer data sets. So we looked at a glioblastoma cell line, and in this case, this glioblastoma cell line is overexpressing ATF3, which is believed to repress tumor suppressor, so that this ATF3 is driving the tumor. And we can then identify regions that are predicted mostly by heterodimer sets. So we look at those regions and did the novel motif prediction with MIM. And in this case, for the peaks better called by ATF3 CBP family, you retrieve the CRICAT site. And for the peaks that are best recalled by this set of heterodimers, we get the AP1 site, which is the preferred site by those heterodimers. And then we did this to see if they differ in terms of geo terms, and they did, not super significant, but they did a bit. And then we look at what's been presented here and discuss a lot. How do you then use your in vitro data to look at the non-coding variants? I kept the old percentage of the Marano. I didn't know we were that high now in 99. So we use this list, the around 5,000 GWAS SNPs. And the way we did it, we just center at that SNP and went 10 bases to each site, and then use a sliding window with our in vitro score. And then we just kept the maximum score for that, to score that allele. And okay, so in this case, it's a prostate cancer risk allele, and you create a CRE site. Yeah, this is with prostate cancer, and then this allele, we chose this. This one has been shown at least by gel shift that allele is differentially bound by CREP1. Okay, if you take the 5,000 SNPs with, in this case, with AP1 dimer and score all, most of them will have no score for that transcription factor. Some of them, the variant will have no effect on binding, but some of those then will be near either axis with potential impact in binding. These two, if you go to ENCODE data, both occur in hypersensitive sites in some line and have bZIP CHIP6 in some lines too. So we did it for our 100 dimers and the 5,000 SNPs. And then we colored blue or red, depending on if we were predicted increase or decrease in binding. And overall, we'll see some variants that we predict to impact a lot of dimers. Some variants we predict to impact just one variant. And then the way we predict this, we do the sliding window, take the maximum, and then we just take the ratio. And we add, we use different constant to add to avoid dividing by the very small numbers, and we just vary that. And in the supplement, we just report like all our scores, whether they are very robust or at least you know when they go away. Okay. So we did DNA binding preference of heterodimers, identified hetero-specific sites, and I think we can improve accounting for CHIP6, including the heterodimer data and the impact of some variations. That was in a SIMS lab. Now in my lab, I just got a grant to study cardiac transcription factors, to look at grammar, using SELEX, to look at grammar of enhancers in cardiac development, impact of missense mutations, and impact of non-coding mutations. But we also got some NSF money to study at red color patterning in butterflies and the gene regulatory networks that drive the coloring in the wings. As Trevor mentioned, then this is my lab. I have 4 graduate students now and mostly undergrads. And then this is how our building looked the next day after the hurricane. And luckily my lab didn't suffer that much. This was in a SIMS lab with this support and collaborating with Amy and Aaron. And I'll be happy to answer.
Questions? Is the butterfly morphology